Good afternoon, everyone. I'm Keith Cook, the David Edward Schramm Professor and Head of Biomedical Engineering. Welcome to the College of Engineering's webinar series, Faculty Insights, a 20-minute briefing. Thank you for being here today. In this virtual series, faculty from across the College of Engineering share insights into their research, its impact, and provide perspective for the future. Following each 20-minute presentation, attendees will have an opportunity to direct questions and comments to the speaker. Our speaker today, Tagbo Niepa, is the Arthur Hammerschlag Associate Professor of Biomedical Engineering and Chemical Engineering. He will give a presentation entitled Engineering Microsystems to Decode Complex Microbial Interactions and Emerging Diseases. The growing complexity of microbial systems, evident in rising drug resistance, antimicrobial failure, and persistent infections, underscores the limitations of traditional tools and the urgent need for innovative technologies in microbiology. In his talk, Dr. Niepa will present the design of advanced microsystems and platforms to address key challenges, including high-throughput cell culturomics, mechanisms of drug resistance, and interrogation of the human microbiome. Together, these approaches enable new ways to study complex microbial interactions and accelerate translational discoveries in health and disease. Tagbo leads a dynamic research lab focused on three key areas: developing alternatives to antibiotics to combat drug-resistant pathogens, uncovering how biofilms form at fluid interfaces, and replicating the human microbiome by encapsulating microbes. His work brings together physical chemistry, biology, and engineering to better understand and control microbial systems. He earned both his bachelor's degree in biomedical engineering and his PhD in chemical engineering from Syracuse University, and completed a postdoctoral fellowship at the University of Pennsylvania. Prior to joining Carnegie Mellon, he served as an assistant professor at the University of Pittsburgh. Tagbo's innovative contributions have been recognized with several prestigious honors, including the NIH New Director's New Innovator Award, the NSF Career Award, and the 2024 Rising Star of Mechanical Engineering Award. Before I turn the screen over to him, I would like to remind our audience to please use the Q&A feature at the bottom of your screen to submit your questions to Tagbo regarding his presentation. You may do this at any point during the program. Nikki Bernardi, Senior Director of Development in the College of Engineering, will be moderating the session, the question and answer session, at the conclusion of the presentation. Tagbo, thank you for being here today and speaking. I'll turn it over to you now.
Thank you so much, Kate, for this great introduction. And I'd like to start by thanking Kelly for organizing this incredible event. Please tell me if you can see my slide.
Looks good.
Perfect. Okay. Good afternoon, everyone. It's such a pleasure for me to be presenting my work and highlight some of the insight that my research group has been developing. I would like to start by thanking my students. Basically, as you can see here on this picture, I'm here with my research group, and those students do an incredible amount of work when it comes to designing this microsystem and trying to understand what microbial dynamics is about. So I'll jump right into my presentation. So here on this slide, basically I see I have one dot somewhere in the middle. I'm sure everybody is seeing that. But the cool thing about that dot is actually a yeast, and that yeast is called Candida albicans. So basically from 1, it goes to 2, from 2 to 4, and just in a matter of 48 hours, meaning 2 days, we went from 1 cells to a billions of cells. So at this point, you can already talk about an infection if you have 1 billion of cells into a gram of your tissue. Candida albicans is actually a pathogen. So in 2019, the CDC put together this report, and the report was actually to highlight the state of infection here in the US. And the report started by this dedication to the 48,000 or the 50,000 family who lost a loved one each year to antibiotic resistance or Clostridia difficile, and to the countless healthcare provider, public health expert, innovator, and others who are fighting back with everything they have. So that report was dedicated to all of us who suffer directly from multi-drug resistant infection, or we're trying to work toward finding a solution to that. If that was about 50,000 families who lost a loved one in the US, worldwide, that was about 1.2 million of people who die from drug-resistant infection. And according to the estimate in 2050, that would be more than 10 million of people that would pass away based on this drug-resistant infection. But when we go deep into that report and check the numbers, we knew that about 3 million of people got infected here in the US, and 50 people pass away. 25% of these death was mostly caused by one microorganism that is called Clostridia difficile. So Clostridia difficile is a drug-resistant infection, a pathogen that leads to an infection, and the CDC referred to that as an urgent threat. So the urgent threat, basically, there's no effective way to manage them today. The serious threat would be those pathogens that constitute the other 35,000 deaths, but then there's some medication for them, but eventually they become more and more resistant to those infections. For instance, I'm listing here, getting back to it, I'm listing here several pathogens such as drug-resistant Candida, Pseudomonas aeruginosa. That's a pathogen that you're going to find in your bathroom, unfortunately. You're going to find Staph aureus. That's a pathogen that you might find on your skin. And Candida auris, which is a new emerging pathogen, also considered by the CDC as an urgent threat. So my lab mission is basically to focus on this type of drug-resistant pathogen and challenging organisms that keep rising and causing some major infections. And what my lab does, my lab design technology in order to eradicate them. We also trying to manipulate fluid interfaces in order to understand how microbes that are relevant to the environment, to health, but also to industry, such as the petroleum industry, for instance, behave whenever they're interacting with different type of fluid interfaces. And my lab focus on designing different type of microsystem in order to explore the untapped potential of many microorganisms that we do not know, because many of them are still considered unculturable. So I'm going to jump briefly into what those research trials are about. For instance, in the first one, where we're focusing on drug resistance infection, we're focusing on designing some type of electrochemical therapy. We're also designing different type of method using nanoparticles, for instance, in order to eradicate drug-resistant infection. So I started my presentation by showing this pathogen, Candida albicans, and that's Candida albicans here being highlighted. In blue, you can see the cell membrane, and the green and the red are basically the shift of a dye called FON1 that shows that this particular pathogen is metabolically active. But my lab designed this technology that is based on electrochemical current. So we're supplying a very low dose of electrochemical current. So a current that you're going to use to supply or maybe to power your device could be here employed, but for a certain purpose, to lyse the cell membrane and make this microorganism much more sensitive to antibiotic. So on the right-hand side, what you see is actually the drugs, fluconazole, which is the most common antifungal drug that has been labeled here with CY5, basically a fluorophore that can now, after the treatment, diffuse into the pathogen and highlight all these target within the cell. So knowing that this type of technology can definitely affect eukaryotic cells, at least lead to one understanding that we can lower the amount of drugs that is used to basically treat those type of pathogen. Recently, we started focusing on Candida auris, which is being considered by the CDC as an urgent threat. Actually, in collaboration with the CDC, we did receive about a panel of 31 strains of Candida auris that were isolated across the country from different patients. And we start testing those Candida auris with 4 major type of antifungal drugs that are used to treat them. And if you pay attention to the color code here, for the drug that is 5-FC or the one that is called fluconazole, basically we have no level of inhibition, even with high concentration of these drugs, meaning that this pathogen is seriously a concern to us. But when we brought that pathogen to the lab and start running some experiment with that, using this technology that involve the delivery of a low-level current, we see that basically the drug that seemed not to enter this pathogen can now easily diffuse into the cell membrane and reach the cellular target, as it was proven previously for Candida albicans. So this technology works in multiple ways. First of all, it does attack the cell membrane by lysing directly microbes, and that's not necessarily the yeast, but also we tested that previously on bacteria. It lyses the cell membrane and allow cellular target to be reached, but sometimes the reaction might lead to some type of byproduct, such as hydrogen peroxide is in this case that is highlighted, but other metabolites that can also diffuse into the cells and lead to the cell killing. Something that we realized in the past is that the reaction can lead to the release of different ions, and those ions can form different complexes with a drug that is present in the system, which might further potentiate the activity of a drug and lead to cell killing. So based on that, my labs are designing different type of technologies. Another focus of my research lab is actually to understand what is happening when bacteria usually interact with fluid interfaces. So thinking about fluid interfaces is basically thinking about what happened when a gecko, for instance, walk on water, or maybe when mosquitoes sit on water. Basically, we can think about that when the organism become extremely small, the organism can interact with this type of fluid interfaces just because of the interfacial tension. Here I have an illustration of what this represents for bacteria. So here you're seeing something that's going to be very similar to observing your glass of water from above. So imagine now on this glass of water, you simply pour a thin layer of oil, and in the water, there was a bunch of bacteria. Usually this is what is happening. Those bacteria emerge, they come to the interface where the oil and the water meet, and then they start forming this type of structure. So the bacteria that were initially single cells now start forming this type of assembly of multiple microorganisms, and that's tend to cover this whole interface with a film that is extremely elastic. So wherever you do have the all-water interface, whether it is, for instance, in milk or maybe during an oil spill, for instance, bacteria could be present, and that's the dynamic that they're going to undergo. They will usually come and be trapped at those fluid interfaces, and they're going to respond by just creating a certain film. And because they can metabolize sometimes some of these interfaces, the film that they're going to make is going to be just a representation of how they feel whenever they see these interfaces. So what I do in my lab, I'm designing some type of droplet, which is a all droplet that I maintain under a suspension of bacteria. And that all droplet could be maintained stable because of the pressure of the needle that is inverted into the oil droplet. So initially, the bacteria will interact with that, and after 24 hours, they will start forming this film that way we can measure the elastical property of this film. So if you want to understand exactly what this experiment looked like, it looked like a balloon that you simply deflate. But here we can inflate and deflate that back just to be able to measure what are the viscoelastic property of a film that those bacteria are making. The microbes here was isolated from a lung of a cystic fibrosis patient, because the lung of a cystic fibrosis patient is a similar type of air-water environment where bacteria gets settled there and they start experiencing this type of dynamics. So if the same microorganism was just cultivated into some type of liquid suspension, they will be just as it is here, basically all spread out. But that particular organism that we saw is called PSL. It's basically for slimy Pseudomonas aeruginosa. These bacteria start making a film, a mucoid film that basically allowed them to have these kind of viscous properties. And then when you put them at the all-water interface, unlike the other type of Pseudomonas that are not necessarily mucoid type, they form an elastic film. So what we realize is that this interfacial energy triggers cells somehow to respond to a certain manner. If they're weak, they could easily be belied by the interfacial stress. But then the cells that know exactly how to adapt to this kind of environment, we start making a film that protect them against the environment. So based on this understanding, my lab is pushing the boundary of the interfacial phenomena when it comes to microorganisms at the all-water interface by studying, for instance, what is happening when those microbes see lotion, which is actually all-water emulsion, or maybe when those bacteria are directly derived or maybe retrieved from a sediment of an oil spill, for instance. We study different type of infection model involving the lung and bacteria involved into those lungs. And even if this can also involve sometimes some viruses, such as the COVID-19 viruses. Basically, our idea here is to see how the interfacial phenomena linked to those microorganisms trigger the response, trigger the formation of the new materials, and further lead to drug resistance. So this is what I signed up for, to talk about some of these microsystems that my lab developed in order to decode a complex microbial interaction in emerging diseases. So whether you're an expert in a field or maybe not an expert, that you just remember your biology course when you were in high school, I'm sure you must have used one of these devices listed here. If you simply a home brewer, for instance, for your beer, you must be using a bioreactor where you simply mix different type of nutrients and microorganisms. But the cool thing about the evolution of microtechnology and nanotechnology is that it allows us somehow to design some platform that basically allow us to look at those microorganisms and how they interact with each one of them, and whenever they form a community. So in number 16 and 17, this is what my lab designed. We designed what we refer to as microcapsule, or again, nanoculture. But we've been inspired by what happened in number 18 because of the fact that most of the microbes are not necessarily cultivable into laboratory conditions. So the one that we can bring to the lab to cultivate and study have been the one that are domesticated, and they're not necessarily those that we find in the wild. So to be able to understand exactly what is happening in the wild, to be able to tap those new microorganisms and see what they produce, basically we start designing a system that's also going to allow us to cultivate those microbes directly where they belong. So our idea here is basically to create a confinement which is going to be semi-permeable, in which we can basically keep those cells, whether bacteria, yeast, and see how they interact with different species that are not necessarily encapsulated with them. But further, what we like to do, basically, we like to use this platform as a mean to directly interrogate those cells with different type of compounds, signaling molecules, antibiotics, and even fluorescent dyes, and be able to tell whenever those cells are physiologically active or whenever they undergo some interaction that inhibit their growth. So we start designing this system that we refer to as the nanoculture. So the nanoculture, they're just a simple suspension of microorganisms into a semi-permeable shell. So in terms of their size, they're microcapsules. That means that they're few micron in diameter. But when you convert that micro diameter into a volume size, basically it brings you to that notion of nanoliter. How do we make them? Basically, we use this type of flow focusing microfluidic system to encapsulate the bacteria into this polymeric shell. So the way it looked like what your kid would do at home by blowing a soap bubble. So the soap represents that shell, and here we use a polymer, and the air that is infused into that shell of soap is going to be actually a bacterial suspension. But outside is maintained spherical just because you do have a surfactant outside that basically allow that phase separation of oil versus water to be kept stable. So by doing that, we can bring those bacteria that has been encapsulated into this polymeric shell to a temperature that is conducive to their growth. Then here I'm performing a Z imaging, meaning that I'm going in a Z direction by just imaging different layers of this microcapsule to be able to reconstruct and make sure that I can understand exactly to what extent those microbes end up going into these microcapsules. So cool thing about the system is made out of polymer. So it means that it's a very short, robust shell, and that shell can last for multiple days. Here, for instance, for 7 days, for 7 months, the shell remain extremely stable. And the way of making that is very much interesting because those capsules, as I've shown here, are very much uniform in their size. But then you can tune the size of those capsules by making them smaller or larger, depending on how you play with the three phases involved. For instance, by doubling or tripling the flow rate of the outer phase, basically, we can make those nanocultures smaller, we can make them faster. And because of their volume here, which is about 2 nanoliter to 5 nanoliter, we refer to them as nanoculture. So we've been making those nanoculture, and because they're made out of a polymer, which is basically lipophilic, meaning it means that multiple drugs will be able to diffuse into that and multiple compounds. So here, for instance, the biofilm tracer that we use to label the cells can easily diffuse in the polymer and stain the cells that have been encapsulated. While we saw evidence that cyto9 can diffuse and stain the cell in green, propionum iodide could not diffuse in. So this brought us the idea of some type of selectivity coming from the type of compound that can diffuse into the nanoculture. And here I'm listing a few drugs. Ampicillin, ciprofloxacin can easily diffuse and inhibit the growth of the cells that have been encapsulated. But when you come to tobamycin, that's not what happened. Tobamycin is a sugar-based molecule, so it means that it cannot easily diffuse into the shell of a polymer, which is a good thing. It means that sugar-based molecule definitely, when they introduce inside, they will remain inside. They will not necessarily diffuse out, which can allow the survival of the cells and allow them to recuperate, to recover and to grow as they're using the sugar as their carbon source. So we did somehow characterize all type of molecule that can diffuse in and out of a nanoculture. And we saw that there's a molecular cutoff, but then came to my lab a student who were very curious about the type of polymer that we're using and wanted to make a new version of that polymer that's going to be much more functionalizable. So what she did was just to mix a ratio of hydride and vinyl, and in the presence of platinum, basically that's how PDMS form. So once she understood that process, she realized that platinum play a very important role in the cross-linking ability of this polymer, but also in its mechanical properties. So another student, Shakira, tested the mechanical property of the composition that Huda, the first student, made, and realize that as you increase the platinum catalyst, basically you make your polymer stronger. And the elastic property that I'm showing here, which is about 600 kilopascal, is very similar of what you will see for the shell of an egg that you have to use every single morning to make your breakfast. So it's very robust. You need something very hard to be able to release that. So that means that once the bacteria sequester into that capsules, it's very hard for them to be released. So once we realize that we can master that chemistry, we can make something that is strong, we start using that actually to encapsulate the bacteria. And here I'm showing a video that has been collected for about 24 hours, where initially 5 cells were seeded, and then those cells continue to divide, continue to replicate into those capsules, and for about 20 hours or 24 hours, we got a capsule that was fully covered with cells. Something cool about the system is that as the cells are metabolizing the carbohydrate and the sugar being present into the capsules, basically they lower the environment, the osmotic environment inside, so water start osmosing out. And this is what is shown here by the change of the size of those capsules. From time 0 to time 20, the capsule somehow shrink because water start leaving the capsule. So this was good. It told us more about the permeability, and we have a long list of stories about how we today use that technology as a mean of continuing to feed the cells as they sequester into those capsules. But as I was showing those different time points, we can only focus on one time point, like the 8 hour. And instead of recording just a picture, we can now record a video and track the cells that are present there. Actually, we've been using this technology as a mean of profiling the growth dynamic of those cells that we're encapsulating so that we can know, for instance, how they displace into the nanoculture and how fast they can go. If there's any trajectory specific to one microorganism, this is something that we use today and develop different type of code to be able to identify those type of cells. So, interestingly, the capsules, they confine and basically closed environment. We can put anything that we want inside. And so one cell, we can have multiple cells. Here, that's ecoculture system in which we have Pseudomonas in green and Staph aureus in red. This was two of the cells that were listed as drug resistant on a CDC list. Basically, we can put them together to see what kind of competition those cells acquire. And as you can see here, basically, Staph and Pseudomonas tend to go nicely into those capsules, but ones tend to outcompete the other. And that's the example of a green cell, Pseudomonas, that outcompetes Staph aureus. Our technology today allows us to quantify exactly how many cells we have into those capsules that we are collecting and we are growing. It's about 44,000 Pseudomonas aeruginosa versus 8,000 Staph aureus. The closer the staph are from each other, the bigger the cluster size, and they're completely surrounded by Pseudomonas aeruginosa. That's what these data show us. So we can perform those type of complex community studies with this type of system. But further than that, what we can do, we can basically quantify the exact amount of cells that we're introducing into those capsules. And the goal for that is us thinking more about how we can use this capsule as a delivery vehicle for cells that are beneficial. And this comes along the idea of treating Clostridia difficile. Clostridia difficile, if you remember from those first data that I show you, the cells that cause about 25% of the death associated with drug-resistant infection. But when somebody has Clostridia difficile infection, the way that the person is treated today is with the fecal matter isolated from a healthy donor. That's what's going to be introduced into this patient so that the bacteria present into that fecal matter can be reseeded into the gut, the GI tract of the infected patient so that it might be able somehow to get rid of the Clostridia difficile bacteria. So what we like to do in our technology using the nanoculture is actually to refine that process because as you can imagine, it's a very messy process. So instead of delivering this fecal matter, what we tend to do in our lab is actually isolating the good germs from the gut of a healthy patient, encapsulating that, which we acquire a US patent for that, and then basically delivering the capsules, this cocktail of beneficial microbes in the gut of a patient, which my student Amy is leading an animal study on. Another vision that we have was actually using the nanoculture as a mean of cultivating soil microbes. So recently, what one of my students did basically was to introduce into the shell of this nanoculture some magnetite. And this magnetite basically can allow to recover those nanoculture when they form. So by putting a magnet next to it, she can pull all these nanoculture from the soil environment. And she tested how much strength it's going to take in terms of magnetic strength it's going to take to recover those capsules. Those capsules not necessarily because they're embedded with some we have some nanoparticle incorporated into the shell of those capsules that they become toxic. Not at all. With the nanoparticles into the shell of the capsules, the bacteria continue to go. If you really want to think about what can kill them, is the environment outside. If you change the acidity of the environment, this is what's going to kill the bacteria inside, but not necessarily the nanoparticles that are present inside. So with that, we can simulate some soil-like environment where bacteria isolated from the soil could be encapsulated, and simply we use a magnet to simply pull all these things down. Right? We have a scenario that is very similar to a soil where we simply use this sand And as you can see, the particles are completely buried into the sand, but then using a magnet, we're easily able to recover these capsules. But I mentioned earlier that the shell of the capsule is extremely strong. That's something that is very similar to the shell of an egg. So how would you remove the bacteria and get the genetic material knowing that this is a very solid environment, robust environment? Well, we did functionalize the capsules, and what we do, we simply introduce them into pure water so that they can swell and release those bacteria that are incorporated on purpose. And this is happening on demand. We can collect those bacteria, we can quantify them, we can see exactly how many are present there. But not only that, we can also extract their genetic material to be able to identify them and then perform any other type of downstream analysis in terms of bacterial classification and identification. Very quickly, I'm going to use the next minute to talk about another type of new microsystem that we start developing in our lab. And this is basically to address one emerging disease that is called babesiosis. I'm sure hearing the word babesiosis may not necessarily sound familiar, as if I was telling you, for instance, malaria or Lyme disease. Lyme disease, many people might be familiar. Malaria is very common. We know that that's a disease that's acquired by a mosquito bite, where a parasite basically goes into the bloodstream. Well, babesiosis is caused by Babesia microti, and in a similar manner that we acquire malaria, but the only vector here, the transmitter here, is going to be a tick. So basically, the tick will bite you and introduce the parasite into the red blood cells, into your bloodstream, and the parasite will find a new host, which is going to be the red blood cells, replicate into the red blood cells to a point where there's no more space, as use all the nutrients, and we egress, meaning that we exit the red blood cells in order to go and attack a new, fresh, new red blood cells. And people who have babesiosis today, the way of identifying that is going to be to perform a blood smear, getting a piece of a tiny drop of their blood, smear it on a glass slide, and be able to detect that. That is not necessarily a high resolution method, so many people could be misdiagnosed, that's for sure. And in addition to that, if you would like to study babesiosis as a disease, you have to rely on a colony of mice, because it's very difficult to grow this germ in a laboratory. And people who have babesiosis are prescribed anti-malaria medication. So, which is funny, meaning that maybe if we understand more about that disease, we might be able to develop much more new drugs that's going to be much more targeting for babesiosis. So, babesiosis is spreading in the US. In Pennsylvania, unfortunately, we don't have enough data to tell exactly how much babesiosis there is here. But at least we can see that in Northeast, that's a disease that is spreading. So there's a strong need for us here, living in Pittsburgh specifically, to care about that particular disease. So my lab started focusing on these questions, and I had a research scientist who joined my lab and basically came up with this new platform where it can functionalize a surface and create a surface that is extremely hydrophilic or a surface that is extremely hydrophobic. So using that platform, basically what he was able to do was just put two droplets of blood together on an hydrophilic surface, and this blood will be somehow resuspended into an extremely hydrophobic surface. So they're going to form those two droplets that will be connected by a bridge, and that bridge in between allow to form a thin layer of red blood cells. So instead of relying on a blood smear, what this allowed us to do today was basically to be able to quantify when babesiosis is present into the red blood cells, as you can see here. The advanced imaging system that we're using in our lab allow us to see different layers of the infection. And when there's one parasite infecting the cells, two parasites infecting the cells versus more than four parasites sometimes, because they do overgo into the red blood cells, this is something that is now visible. We can go and further characterize those red blood cells and see that by using this dye, the PKH67, basically we can highlight all the phospholipid, including the phospholipid of the parasite whenever it's located into the red blood cells and infecting the red blood cells. And the TEM and the SEM on the right-hand side show where the parasite end up locating into the red blood cells, which deform them entirely. The last slide I'm going to show you is actually a cool event that we found out about that parasite that basically revealed a new paradigm when it comes to the infection process of those parasites. In the past, we simply thought that they're going to be introduced into a bloodstream by that tick bite and then go to red blood cells, divide, and basically go and trying to find a new host. But now something that we realize is that the parasite can even move the red blood cells. So this looked more like hijacking a car to flee away from the police because around the parasite, there's usually some white blood cells that are looking for these type of infections and trying to basically um, uh, stop the infection or attack the parasite. But now with this type of new microsystem, this type of new platform, we're able to reveal unique knowledge that weren't known before. So the type of microsystem that we're developing in our lab basically allow us to have a better understanding of microbial dynamics at the single cell level, but also within the community. It also allows us to basically review new paradigms such as this regarding this parasite. But if we like to test a new type of compound, now we don't have to worry about the volume of this compound. They could be even in a small amount and still be tested and reveal how effective they are regarding those emerging diseases. And furthermore, we've been using this as a mean of designing some type of culturomic approach that will not necessarily allow us to rely always on mice in order to be able to detect those type of parasite or infectious diseases. With this, I would like to acknowledge my research group. I have a great set of brilliant students that basically push the boundary of our science, and we're very much excited of every single new knowledge that those students discover. And in addition to that, I would like to thank all the collaborators and mostly the funding sources that allow us to do this work. Thank you.
Thank you, Tagbo. We very much appreciate all of these great insights you've shared with us today. We will now turn to our audience for questions. As a reminder, please submit your questions via the Q&A feature at the bottom of your screen. Make sure to include your name with your question. Okay, so Tagbo, the first question I have is, how long until this research can help in the healthcare field?
That's a great question. Again, for research to be able to move forward, I think everything depends, of course, of the level of funding that there is to allow us, for instance, to achieve all these goals. With what we have so far, I think we start revealing some new interesting knowledge. And at this point, for our next goal, what we are aiming for is actually to develop some type of automated culturomic systems that at least enable, for instance, for the last story, to be able to cultivate those cells that are extremely challenging to maintain outside of a mice system. Regarding the microbiome study, especially with encapsulation, the study of a microbiome such as that of the oral environment involved around 700 species. To be able to study just 5 species, it's about 30 experiments. So if you imagine how long it's going to require to study about 700 species together, this is going to require much more automation. And this is where we're moving toward. We hope that in the next 5 years, we're going to be so much advanced that we'll be able to reveal new knowledge regarding the human microbiome and deciphering, for instance, what are the main key players into the gut microbiome, which could be applied toward Clostridial difficile infection.
Wonderful, thank you. This question is from Hillard. Fantastic work. Can your electrical work on CRS be used for drug discovery? In other words, is there a drug that mimics the electrical effect on the cell membrane? And is the target a membrane pump, pore, etc.?
I'm not sure about the target of the electrical treatment. If this is a pump or the flux system of the yeast, it's not clear yet because we haven't performed any transcriptional analysis, for instance, to determine based on genes what gene get overexpressed. But what we realize is that the cell membrane might be severely affected. Some basic SEM and TEM pictures that we perform so far have shown that there's some major disruption on the cell membrane. So I'm not sure the fact that the electron and the ion are moving fast into the field if this is really if disrupting the cell membrane is the main target here. It's more likely that this might happen. But previous study has shown that you can use electrical field somehow to open and close different efflux system, including the pore that the cells might have. I'm not sure if this is going to be applied and if this is related into that particular context. The second question regarding any type of drug that might imitate the electrical field. Yes, we're aware of a few drugs that can cause some type of membrane depolarization. And that's my strong belief that anything that might somehow lead to membrane depolarization might somehow be effective in terms of introducing and permeating multiple biocide into the cells. But we haven't explored those type of drugs yet in this context.
Great, thank you. This question is there an upper limit in the number of cells you can encapsulate in these nanospheres? Does this limit depend upon the oxygen levels?
I think the limit will depend mostly on the volume of the cells and the amount of carbohydrate and metabolite or nutrients that you have into the cells. So it end up being a mass balance problem. So based on the volume of these capsules, you can encapsulate almost like millions of cells if possible. That means that if you were just to get those cells and pack them and squeeze the capsule, you can get much more than what is currently encapsulated. The only thing is the amount of sugar that we use has to be so that it does not necessarily lead to any other type of cell drying or maybe because of osmosis and lysing the cells. So what is a safe level of carbohydrate that has to be introduced and how this carbohydrate is going to be metabolized into new cells is that mass balance problem that we have to take in consideration in order to get the maximum yield into those capsules. So that's a great question.
Great, thank you, Tagbo. This question is from Ned. Is this similar to electroporation for getting DNA into cells?
Very much similar, I would say. The only difference here is just that electroporation, you're going to deal with a lot of a large voltage, like 1.5 kilovolt, for instance, is what allow you to have that pulse in order to introduce genetic material into DNA. But what we're performing here is actually a treatment with very low dose of current. So the voltage, whenever effective, end up being about 5 volts. That's the maximum voltage that we ever achieve. But for a current density that corresponds to 70 microampere per centimeter square. So it means that that current density is below the perception level of human. And we apply that for about an hour. And this is how basically we start achieving this type of killing mechanism. So it's not quite similar because electroporation, it can require much more voltage compared to the treatment that we are applying here, which is performed at low voltage over an hour.
Wonderful. What sources of grants and funding support your research? For example, NIH, CDC. This question is from Ed F.
Can you repeat that question again?
What sources of grants and funding support your research? For example, like NIH, CDC, et cetera.
So my research has been supported by NSF and NIH so far. And again, my research personnel, including my students, have been supported by different type of foundation mechanism. Yeah.
Okay. So are you working with any of the medical centers here in Pittsburgh?
If I'm affiliated to a medical center, is that what you say?
Yes. Are you working with any medical centers here in Pittsburgh?
I do have a collaboration with the medical school. So for the Babesia project, we do have a collaboration with the School of Public Health. That's where my collaborator, Daniel Toff, is. When I was back at Pitt, I was interacting much more with the Center for Microbiome and Medicine. And I'm currently collaborating with the Pitt Dental School. So we do have some collaboration. And most of the study that we do regarding NMR, for instance, to determine what the composition of the EPS that the cells are making at the all-water interface are performed at the NMR facility in medical school.
Wonderful. So what's the most surprising thing that you've learned about bacteria through your research?
The most surprising thing that I've learned about bacteria is bacteria are much more of a mystery. They're much more beneficial than we can expect from them. It's more likely that whenever we hear the word microbes, everybody will tend to stay away because we think that they're all bad. But actually, the population of the diversity of microorganisms that are pathogenic is much lower than the population of microorganisms that are beneficial. So the most interesting thing that I've learned is that just 5%, according to the estimate, are culturable. So that means that there's 95% that we do not know of microorganism. And if you think about the way that penicillin was discovered, usually we say by accident, just because one microbes go next to a pathogen and release some type of toxic compound that that were actually extracted later on and characterized as antifungal and antibiotics. It's basically the way that we continue to discover most of these drugs today. It's true that today with the rise of AI, it is possible to think of the DNA of the cells and trying to see if they make any kind of toxin that can serve potentially as antibiotic. But microbes has a key to so many questions that currently we're asking. And if today we even think about the vaccine, the COVID vaccine, for instance, um, the whole design of that medication is linked to microorganism as well. Like some bacteria being extracted from uh, um, the Yellowstone, for instance, where they live in extreme environment, can reveal so much information about how we should treat different diseases today. So there's a lot to know about them, and we still started.
It's fascinating. I have a comment here from one of our attendees. In the comment is, I don't think that only carbohydrates are the limiting factor. I think available oxygen will be much more important as it will deplete much faster. And they shared a paper, so
Okay.
I can
Yeah,
send that to you, Tagbo.
I like the comments, and I didn't quite list oxygen and all the gases because of that exact reason. And uh, the polymer is permeable to gases. So if you think that oxygen is a limiting factor, you can simply supply oxygen to your system and the gas is not to diffuse in. So PDMS has no limitation with gases. So that's the reason that I was worried more about the nutrient content. So I'll be more than happy to talk to that person specifically because gas are not a limiting factor in terms of how they diffuse into the polymer.
Great, thank you. This question is from Mary. How would the electric current be applied in humans with resistant pathogens, and could it affect non-target cells?
That's a very good question. And Mary, in past 5 years, the Department of Defense has been focusing on using this type of technology and further developing that. So ARPA-H, for instance, as you call, and basically this technology has been much more advanced that we could present here. But the idea is that electrical current has been used already in the medical field, sometimes to perform a surgery, sometimes to measure different type of signal from a body or just introduce those signals in the body. So the way that we envision that is going to be to start designing some type of electrochemical bandages, which my lab has already started, and we're moving more towards some type of animal studies. And because the current that is delivered is very low dose, it would not necessarily lead to any electrocution of a patient. It's going to be much more localized to the site that has the infection in order to eradicate the infection. So this is something that is worth trying, and this is the reason that the funding agency currently is supporting work like that, including ARPA-H.
Wow, very interesting. This question is actually from Keith Cook. One of the oldest historical antibacterial materials is copper. Do you know the cause of its antibacterial effect? Is it similar to the electrostatic interaction?
I think copper, the idea might be much more you may want to correct me if I'm wrong, much more linked to depolarization of a cell membrane. So I'm not quite sure, but copper has been used as far as we know as an antimicrobial. That's the reason that most of the doorknobs was coated with copper, for instance, right? But the way that copper works is going to be completely different from the way that this electrochemical treatment works, because the electrochemical treatment generating electrical fields, you have some current density that is basically mediated throughout the electrolyte. So you have a presence of ions, reactive oxygen species. You have a direct effect on cell membrane that lead to depolarization. So it is a mixture of much more effect that lead to the killing that we're observing using the electrochemical treatment of microorganisms, compared to the static effect that copper, because interacting with cells, might have and lead to local depolarization and then killing.
Thank you.
Okay, is this type of research typical in a chemical engineering department, or is it unique to CMU's College of Engineering, and why?
That's great. That's a great question. Most of the research that we're doing multidisciplinary and interdisciplinary. So you might see research of that nature involving electrochemistry that might fit into a chemical engineering department. The second research that I've shown that was much more based on bacteria interfacing with all water, that's colloidal science, so it's also part of chemical engineering. The design of a polymeric shell to make those nanoculture is part of chemical engineering, because this is also, of course, polymer engineering. And the design of this type of micro channel using all water interface on functionalized surface are also part of chemical engineering, but they're much more disciplinary, interdisciplinary, so that they sometimes sound as if, well, it's still microbiology or it's still chemical engineering. But basically, that's the nature of research today, moving different boundaries. And we use all physical knowledge, all basic knowledge, and apply that to the solution of different problems. And I think that's what engineering is.
Okay, we have time for one more question, Tagbo. Why do antimicrobial therapies continue to fail against persistent and biofilm-associated infections despite extensive drug development?
So most of the antimicrobial today continue to fail because the microorganism um, biological entity that can basically respond to whatever attack they are under. So they adapt to their environment and basically change, they mutate, and they can respond to anything that's going to be toxic to them. So we refer to that as microevolution, the fact that they can change because they've been exposed to different type of toxic environment. So as microorganisms will continue to have genes, and genes will come from their DNA. The fact that they do have a DNA, those cells will always develop some type of resistance. Sometimes the resistance can develop even before the drug is fully discovered and fully approved by the FDA, because this is a process that happens very fast for them. So that's the reason that in the core of my lab, we do believe that we have to rely on physical chemical events that may not necessarily rely on the cell metabolism, on the DNA factor, the fact that the cells can mutate, to be able somehow to lyse the cells and lead to their killing if we need to find strategy to eradicate drug-resistant infection. So we want to use a drug as a support, knowing that they can also face that resistance, but use different type of physical chemical factor to induce the effectiveness of, or to promote the effectiveness of the drugs.
Very, very fascinating. Thank you again, Tagbo, for a very thought-provoking and informative program. I would also like to thank our audience for participating today. I hope you will be able to join us at our future Faculty Insights programs. Please keep an eye out for a follow-up email soon, which will include a link from this session. Thank you again, and have a great day.